Hello students, in the previous video we studied about fat metabolism, that is by beta oxidation pathway. In this video I am going to explain you about the alternate pathway of glucose breakdown, that is PPP pathway. PPP pathway or pentose phosphate pathway, which is also called as HMP pathway, that is Hexose monophosphate pathway. Okay. So actually we studied about the breakdown of glucose through aerobic respiration, in which glucose breakdown occurs through glycolysis, link reaction and Krebs cycle. But here direct oxidation of glucose occurs where there is no glycolysis, link reaction and Krebs cycle, and hence this pathway is also called as direct oxidation pathway, where glucose oxidation occurs directly without involvement of glycolysis, link reaction and Krebs cycle. And this pathway is also called as glycolytic shunt. And it is also called as HMS pathway, Hexose monophosphate shunt. And it is also named after the names of the scientists who discovered this pathway, that is Lippmann, Warburg and Dickens cycle. This pathway occurs in both prokaryotes and eukaryotes, and it occurs in both the cytoplasm and chloroplast. It occurs inside the cytoplasm of prokaryotes. In case of eukaryotes, it occurs inside the chloroplast in the presence of oxygen. Chloroplast, that too in the presence of oxygen. And this is the only pathway in which, only respiratory pathway in which instead of NADH, NADPH is synthesized. So instead of NADH, NADPH is synthesized during this pathway. And the substrate for this pathway is glucose 6-phosphate. Glucose 6-phosphate. Now let's study the details of this pathway. What happens during this pathway and how glucose breakdown occurs through this pathway directly without occurrence of glycolysis, link reaction and Krebs cycle. Right. So now this is one molecule of glucose. Okay, let's study this pathway for 6 molecules of glucose. So 6 molecules of glucose, that is 6 carbon sugar, undergoes activation into 6 molecules of glucose 6-phosphate. Here 6 molecules of ATP is consumed, and this step is catalyzed by the enzyme thiokinase. This step is catalyzed by the enzyme thiokinase. Right. Now the 6 molecules of glucose 6-phosphate undergoes dehydrogenation or oxidation into 6 molecules of 6-phosphogluconolactone. So the step is catalyzed by the enzyme glucose 6-phosphate dehydrogenase. So what is happening here? Dehydrogenation or oxidation is taking place, due to which from the substrate, that is glucose 6-phosphate, protons and electrons are released, and now these protons and electrons which are released from this substrate are picked up by NADP+, so that NADP+ is reduced into NADPH plus H+. Okay, now these 6 molecules of 6-phosphogluconolactone undergoes hydration into 6 molecules of 6-phosphogluconic acid. So this hydration step, this reaction is catalyzed by the enzyme lactonase. So here this is lactone. Lactone means it consists of keto group. So to this keto group, when water is added, what happens? It will be converted into acid, and hence the 6-phosphogluconolactone has been converted into 6-phosphogluconic acid due to dehydration, which is catalyzed by the enzyme lactonase. Right. Now the 6 molecules of 6-phosphogluconic acid undergoes oxidative decarboxylation. So as decarboxylation occurs, carbon dioxide is released. As there are 6 molecules of carbon dioxide is released, and as it undergoes oxidation, protons and electrons are released. Now these protons and electrons are picked up by NADP+. So NADP+ is reduced into NADPH plus H+. So 6 molecules of NADPH plus H+ is formed during this reaction. And this reaction, that is oxidative decarboxylation, is catalyzed by the enzyme 6-phosphogluconate dehydrogenase. so that it will become, this substrate is converted into, what is this substrate? 6-phosphogluconic acid is converted into now 6 molecules of RUMP. So this 6-phosphogluconic acid is 6 carbon compound. Now it has been converted into 5 carbon compound because here carbon dioxide is lost. That means 1 carbon is lost. 1 carbon from 1 molecule of 6-phosphogluconic acid. 6 carbons from 6 molecules. So here 6 into 6 molecules into 6 carbons. How many carbons? 36 carbons. 36 carbons. Here 6 molecules into 5 carbons. So how many carbons? 30 carbons. So 36 minus 30, 6 carbons are lost. That means 1 molecule of glucose 6-phosphate has been broken down by the end of this oxidative decarboxylation. So now the 6 molecules of RUBP undergoes a series of reactions, undergoes regeneration into 5 molecules of glucose 6-phosphate through a series of reactions. So during this regeneration, different types of substrates are formed, intermediates are formed. These intermediates are erythrose 4-phosphate, xylulose 5-phosphate, ribose 5-phosphate, and then pseudoheptulose 7-phosphate and fructose 6-phosphate also. So 4 carbon compound, 4 carbon sugar, 5 carbon sugar, 5 carbon, 6 carbon sugar, pseudoheptulose 7-phosphate is 7 carbon sugar. So all these types of intermediates which are formed during the regeneration of RUMP into glucose 6-phosphate can be utilized. These intermediates can be utilized by the cell as precursors for the synthesis of other biomolecules required by the cell as per the need of the cell. Okay, now these 5 molecules of glucose 6-phosphate. So this is 6 carbon compound. So 5 molecules of 6 carbon compound is nothing but how many carbons? 30 carbons. So here also 30 carbons. But RUMP, that is ribulose monophosphate, has been regenerated into 5 molecules of glucose 6-phosphate. So by the end of this oxidative decarboxylation itself, 1 molecule of glucose 6-phosphate has been broken down. That means 6 carbons have been broken down. So here, now these 5 molecules of glucose 6-phosphate can join with 1 more molecule of glucose 6-phosphate to become 6 molecules, and this cycle can be continued. Now this PPP pathway is also called as amphibolic pathway because it involves both catabolism and anabolism. It involves catabolism because during this pathway glucose 6-phosphate undergoes breakdown. So glucose breakdown is taking place. And it is called anabolism. It involves anabolism because the intermediates which are formed during this pathway are utilized as precursors for the synthesis of other biomolecules required by the cell. So it also involves anabolism. As it involves both catabolism and anabolism, it is also called as amphibolic pathway. Amphibolic pathway. Right. Now let's see the energetics of this pathway. Energetics, that means how much amount of energy is released by the breakdown of 1 molecule of glucose 6-phosphate. So when 1 molecule of glucose 6-phosphate undergoes breakdown through this pathway, what is happening? NADPH plus H plus are released. The energy is released in the form of NADPH plus H plus. How many molecules? Here 6 molecules and here 6 molecules. So totally 12 molecules of NADPH is formed. So when 1 molecule of glucose 6-phosphate undergoes breakdown, 12 molecules of NADPH plus H plus is released. So when this NADPH plus H plus undergoes breakdown through ETS, how many ATPs will be formed? Just as in case of NADH, even NADPH also produces 3 ATP when it undergoes breakdown through ETS. So 12 molecules of NADPH plus H plus into 3 ATP. So how many ATPs will be? 36 ATPs. So 36 ATPs will be formed by the breakdown of one molecule of glucose 6-phosphate. But the main aim of this pathway is not the production or the synthesis of ATP. Its main aim is to synthesize NADPH plus H plus, which are required for reduction reactions that occur inside the cell. And the other aim is to provide intermediates. Which are required by the cell for as precursors for the synthesis of other biomolecules inside the cell. So this is all about PPP pathway. Hope you understood this PPP pathway, and in the next video I will be explaining about the significance of this PPP pathway, that is pentose phosphate pathway. Thank you, students.